Hello, in this session of biochemistry, we will be learning about enzymes and the concept of activation energy and transition state. The enzymes of a cell are truly a wondrous collection of miniature molecular machines. Enzymes are mediators of metabolism, responsible for virtually every reaction that occurs in a cell. Without enzymes, metabolic reactions would proceed so slowly as to be imperceptible. The term enzyme is generally reserved for protein catalysts, while the term ribozyme is used for RNA catalysts. Even though enzymes are proteins, many of them are conjugated proteins. That is, they contain non-protein components called cofactors, which may be inorganic like metals or organic. Vitamins and their derivatives often function as coenzymes. Cofactors are important participants in the functioning of the enzyme, often carrying out activities for which amino acids are not suited. For example, in myoglobin, the iron atom of a heme group is the site where oxygen is bound and stored until required by cellular metabolism. The properties of enzymes. Enzymes are required only in small amounts. They are not altered irreversibly during the course of the reaction, and therefore each enzyme molecule can participate repeatedly in individual reactions. They have no effect on the thermodynamics of the reaction. Let's discuss more about the third point. Enzymes have no effect on the thermodynamics of the reaction. This point is particularly important. Enzymes do not supply energy for a chemical reaction and therefore do not determine whether a reaction is thermodynamically favorable or unfavorable. Similarly, enzymes do not determine the ratio of products to reactants at equilibrium. These are inherent properties of the reacting chemicals. As catalysts, enzymes can only accelerate the rate at which a favorable chemical reaction proceeds. Enzymes are highly specific. Enzymes are highly specific in the reactants they can bind and the reaction they can catalyze. The reactants bound by an enzyme are called substrates. For example, if the enzyme hexokinase is present in solution with 100 low molecular weight compounds in addition to its substrate, that is glucose, only the glucose molecules will be recognized by the enzyme and undergo reaction. This type of specificity, whether between enzymes and substrates or other types of proteins and the substrates that they bind, is crucial for maintaining the order required to sustain life. The activity of enzymes can be regulated to meet the particular needs of the cell at a particular time. Enzymes typically increase the velocity of a reaction by many folds. They accomplish this feat at the mild temperature and pH found within a cell. The concept of activation energy and overcoming the activation energy barrier. How are enzymes able to accomplish such effective catalysis? Chemical transformations require that certain covalent bonds be broken within the reactants. For this to occur, the reactants must contain sufficient kinetic energy that they overcome a barrier called the activation energy. That means the reactants must have sufficient kinetic energy to overcome the activation energy barrier. Now let's have a look on this diagram, which represents the activation energy by the height of the curves. The reactants in a chemical reaction are often compared to an object resting on top of a cliff ready to plunge to the bottom. If left to itself, the object in all likelihood would remain there indefinitely. In this case, no enzyme or catalysts are in action. However, if someone were to come along and provide the object with sufficient energy to overcome the friction or other small barrier in its way and cause it to reach the edge of the cliff, it would spontaneously drop to the bottom. That means if catalysts or enzymes are in action, the object will reach to the edge of the cliff and it would spontaneously drop to the bottom. It shows that the object has the potential to fall to a lower energy state once the kinetic barriers have been removed. The concept of transition state. When the reactants are at the crest of the energy hump and ready to be converted to products, they are said to be at the transition state. At this point, the reactants have formed a fleeting activated complex in which bonds are being formed and broken. Unlike the difference in standard free energy for a reaction, the activation energy required to reach the transition state is not a fixed value, but varies with the particular reaction mechanism utilized. Enzymes catalyze reactions by decreasing the magnitude of the activation energy barrier. Unlike catalysis by heat, enzymes cause their substrate to be very reactive without having to be raised to a particularly high energy levels. How the enzymes are able to lower activation energies? Enzymes are able to lower activation energies by binding more tightly to the transition state than to the reactants, which stabilizes this activation complex, thereby decreasing its energy. The importance of transition state. Compounds that resemble the transition state of a reaction tend to be very effective inhibitors of that reaction because they are able to bind so tightly to the catalytic region of the enzyme. Antibodies normally do not behave like enzymes, but rather simply bind to molecules with high affinity. However, antibodies that bind to a compound that resembles a transition state for a reaction are often able to act like enzymes and catalyze the breakdown of that compound. What happens after the transition state? As the transition state is converted to products, the affinity of the enzyme for the bound molecules decreases and the products are expelled.